place where you can. So yeah, the first search we used ViewPathDB, which is a place where you can leverage information across species, right? So you can ask a question in Cryptosporidium and then look for the orthologs in oomycetes, for example, or or go look at genes that are have a particular profile in Saccharomyces, and then see if they have homologs in Plasmodium, because you're interested in whether they are some cell cycle regulated genes in Plasmodium that are homologous to the yeast ones. And so you can do this in ViewPathDB. It's pretty nice. There are some short.
Are you sharing your screen? We just see a landscape.
Aha. I'm sharing the wrong screen. That is the problem. So let me do that.
Yeah, we got the landscape again, but it's pretty.
Yeah,
it's a pretty landscape, right? Yeah, I was just showing the homepage, the ViewPathDB homepage anyway. So yeah, there are some limitations, however, in ViewPathDB, because there are certain searches that are only available on the component sites and not in the main site. That's for technical reasons, which we won't go into the details, and hopefully will be resolved, but it won't be soon. And so you have to be aware that there are certain search types that are only available in the component sites. And as an example, if I go to CryptoDB or ToxoDB even, or even TriTripDB now, there are searches that allow you to find genes based on subcellular localization. And so here I am in, let me look here, or gene list, maybe lists of genes. So for example, there's this protein targeting and localization gene lists in CryptoDB, right? And so I can go here and I can ask a question about this. This search is not available in, as far as I know, not available in ViewPathDB, but I may be interested in knowing, this is a real experiment where they looked at the subcellular localization. I may be interested in knowing how many of the rope tree proteins have homologs in Cryptosporidium, have homologs in Plasmodium, for example. So here are these 24 genes, but I want to find the homologs in Plasmodium of these genes. And it's super hard to do this here. Well, you can't do it here in CryptoDB. I can take these IDs and download them and then go to ViewPathDB and run an ID search. But we provided a shortcut. So I just want to make sure people are aware of this. So I can click on this button here and say send to ViewPathDB. And when you do this, it's going to send the same, it's basically sending the gene IDs to ViewPathDB. And so once they arrive here, and you can do this at any moment, like, so you could be running searches in Plasmodium and you think, oh, I wonder if there are any homologs in fungi and whatever, Aspergillus for whatever reason, right? And so you can then send your strategy to ViewPathDB, and then you can add a step here, and I can say, okay, transform to orthologs. And in this case, I'm interested in, let's say, Plasmodium falciparum 3D7. And so I'll pick that and run the search. And so it turns out, there are 7 genes in falciparum that have, there are 7 Cryptosporidium genes that are localized to the rope trees that also have homologs in Plasmodium. And you'll see that there are a bunch of them that are rope tree proteins as well. And so that's interesting, right? So I would certainly recommend that you use that tool. It's quite useful. The second thing I wanted to mention about the Go enrichment, and we were talking about this in my breakout session, and maybe it came up before, but with any kind of enrichment analysis, you should think about how well your background, your reference genome is annotated with whatever terms you're enriching for, right? So if you have a genome that has 5,000 genes in it and only 500 of them have Go terms, right? Then your Go enrichment is going to be very limited. Your analysis is going to be very limited. So it's always good to say, well, to think about your genome of interest and whether it's well annotated for whatever entity you're enriching for. And with the example we did with Plasmodium, let's see, maybe I can do it again here. Oh, I have it here. So you can run a Go term search. I won't replicate it here just for the interest of time, but you can run a Go term search here and see how many genes in the genome have a Go term. And so it turns out in Plasmodium falciparum 3D7, there are close to 4,500 genes. And there are about 5,700 genes in the genome. So it's a very good percentage of your genome that are annotated with Go terms. And so it's a good thing to keep in mind. And so if you do metabolic pathway enrichment, which we have a tool for metabolic pathway enrichment, you should ask, well, how many genes in my genome that I'm looking for enrichment in actually have EC numbers, because these are based on EC numbers, have EC numbers. Is it good? Is it bad? I'm only looking at a very small subset. And so those are things you should keep in mind as you do this. And the last thing is we talked about this word enrichment. So this word enrichment is again just the same statistical test that's being applied, but instead of applying it to the ontology, the Go ontology, gene ontology, it's being applied to the words in the product descriptions of the genes, right? So so that's useful sometimes because you could have a large expanded gene family in your species of interest that has a specific name, like whatever, I don't know, Leishmania, invasion proteins, right? And you can imagine there are like 1,000 of them, whatever, in the genome, and they may not have a Go term associated with them because there is no Go term describing that function yet. And so as a result, you'll never find these in your Go enrichment analyses, but those may come out if it turns out your results are enriched for that. Then you may find them out in this. So I always recommend that people run this. You'll find junk in there too, because like the word protein, if you have a bunch of proteins that are called hypothetical protein, right, then the word hypothetical and the word protein will come up as enriched, right? So you just have to sort of mentally filter those out, even though sometimes the word hypothetical being enriched could be a useful term to sort of say, well, it turns out the majority of my genes that are enriched are hypothetical, and then move on from there. Okay, that's all. Yeah, go ahead, George.
Just a question. The first box in your last strategy, the Go term 4,481. How did you get the number 4,500?
Yeah, so I can repeat that search. So I just go into searches, search for Go, the Go term search, and then you can go to the Go term search. What I did, I filtered on 3D7 because that's the one I was interested in.
All right, okay.
And then I went down to the text search and I just entered an asterisk for the wildcard, and that returns any gene with any Go term, basically.
Great, thanks.
And in fact, you can use this anywhere. So if let's say you want to find all genes, everything in the database, you can put an asterisk up here. It may take a while depending on the database, but you can do this and get all text results in the database, right? Yeah, so anywhere we have a text box, you can use where you can use these wildcards. You can use the wildcard alone to find everything.
Great, thanks.
Sure. And then maybe I'll end here quickly by saying that we have this search that allows you to upload your set of IDs, because in the group we're talking about, somebody was talking about their proteomics experiment, and I was like, yeah, you can take, for example, and they were already doing that, which is great, but you can take, for example, your list of 200 gene IDs or 300 gene IDs that came out from your experiment that seem to be doing something interesting, and then you can just go and run the search for a list of IDs. There's one ID in here, but you can have 1,000 or whatever you want in there. And once you run it, it's going to start a strategy with your list of IDs in there. And now you can do Go enrichment on them, or you can add steps and ask, well, how many out of my 300 genes, how many are also expressed in gametocytes, or how many have a transmembrane domain? You can start asking these questions in a strategy based on your applying those results to your gene list. And I will stop here.
That's great. Yeah.